Um, Hello, everyone. We're Group 1, and we're Ayushi Bomiredipalli, Lily Dines, Daniel Aragon, Isabel Arredondo, and Shreya Bhardwaj. Our group today is going to be presenting on the hepatitis C virus and the interferon therapy that's used for its treatment. Next slide, please. A little preview of what we're going to talk about. We're going to talk about the background of the Hep C virus that's going to help us understand the interferon therapy that's used for its treatment. We'll then talk about the side effects of this treatment and some alternative treatments that could be useful. And then we'll talk about references that we used to get our information. Next slide, please. A little background about the HCV virus and its structure. HCV is a member of Flaviviridae, which is group 4 virus of the Baltimore classification. It's a positive single-stranded RNA virus that replicates within the cytoplasm and is enveloped. It uses viral RNA-dependent RNA polymerase and is highly genetically diverse, like the HIV, because of high error rate of RDRP that creates a lot of mutations. It's a diverse virus with a small genome of about 9,600 nucleotides, and it's a member of the genus Hepasivirus within the family Flaviviridae. This genus also includes pestivirus, pegivirus, etc. And all of these viruses that fall under Flaviviridae are similar in genome organization and have overlapping aspects of viral replication. Now, viral entry. So HCV virus uses receptor-mediated endocytosis to enter the host, which is generally liver cells or hepatocytes. The hepatitis C virus attaches to 3 different host receptors initially, which are HSPG, LDLR, and SRBI. Out of all of these, SRBI is, I think, the most interesting because, and it's also really important because it induces a conformational change that exposes a binding site called CD81. And this CD81 exposure is really necessary for the initiation of endocytosis. Once the virus enters a cell inside the endosome, low pH acts as a triggering signal inside the endosome, which triggers fusion of the viral membrane with the endocytic membrane, and that causes all of the viral genetic material to spill out into the cytoplasm of the host, which are generally liver cells. I'll now pass it on to Lily, who will talk about viral replication.
Hi guys. Okay, so as Ayushi said, this is a group 4 virus, and transcription is done by viral RDRP, going from positive single-stranded RNA to negative single-stranded RNA to mRNA. Translation is where it gets a little bit trickier. The HCV virus has something called the IRES, or the internal ribosome entry site, in its 5' non-coding region, which first binds the 40S ribosomal subunit. This complex then recruits the eukaryotic initiation factor 3 and the ternary structure complex with methionine tRNA, eukaryotic initiation factor 2, and GTP. These all converge and then transition to the ADS ribosomal subunit where translation actually is initiated. As far as proteins that are formed, excuse me, one large polyprotein is formed at first, and then it is cleaved into 10 smaller proteins. The last 3 proteins that are created are structural proteins that surround the nucleic acid, and the last 7 are non-structural proteins, including an ion channel. Next slide. So now we'll talk about the symptoms of HCV infection. The virions primarily infect liver cells, and up to 10 to the 12 viruses are produced per day, which is a crazy amount. It's actually 100 times more than HIV. So as far as acute symptoms go, basically all the symptoms of liver damage and jaundice, and it happens. It starts about 2 to 12 weeks after the HCV virus enters the body, which is, a pretty long time as far as viruses go. But chronic long-term symptoms include fibrosis, cirrhosis, and hepatocellular carcinoma. Fibrosis being light scarring of the liver, cirrhosis being heavy scarring of the liver and the most common form of liver disease, and hepatocellular carcinoma is actually liver cancer. Next slide. The issue with these symptoms is that they develop really slowly over a long time. So many people don't even know they have HCV for years until they end up finding out they have severe liver damage. So that's why treating HCV is important. Um, so obviously HCV treatment would slow or stop liver damage, which would do a lot of good things. It would reduce the need for liver transplants, reduce medical costs, increase the quality of life of patients, and increase life expectancy. As you can see in the picture, up to 85% of people with HCV have the chronic form, and about 20% actually develop liver damage, which is a really scary thing. So now I'll hand it over to Daniel.
So a brief review of interferons is that interferons are a group of signaling proteins made and released by host cells in response to the presence of a PAMP. When a PAMP is detected by the cell, the virus-infected cell will release interferons, which would cause nearby cells to heighten their antiviral defenses. So the production of interferons would allow for communication between infected and non-infected cells to be able to communicate, to be able to stimulate the production of ISGs, which would provide defense against the virus. And these ISGs are genes that make antiviral proteins, which can target different stages of the virus's life cycle, preventing the virus from replicating. And so here's a diagram of the IFN pathway, which we know that there are two arms of the IFN response, which is the IFN production and the IFN signaling, which leads to the stimulation of ISGs. Next slide.
Yeah, so I'm going to talk about HCV and interferon a little bit more specifically. So the main way HCV is detected is by TLRs, specifically TLR3, which binds to double-stranded RNA during the replication stage of HCV. And this uses the MID88 signaling. And then there's a downstream signaling, which includes RIG1 activating MAVs. And as we learned, that is attached to the mitochondria. And this induces the production of interferons. And then finally, we have the binding of interferons to IFNAR at the membrane, which leads to the production of ISGs through the JAK-STAT pathway. Next slide, please. HCV has actually developed multiple ways to evade the interferon signaling pathway. Just to list a couple, the HCV NS34A protein, which is boxed in purple, helps with the maturation of NS proteins, but also cleaves MAVs and TRIF, which inhibits the interferon production. There's also an NS5A protein boxed in red, and this prevents the pathogen from being identified and attenuates the TLR signaling pathway, reducing cytokine production and interferon production. And then finally, there's the NS4B protein boxed in indigo, and this interacts and sequesters STING with TBK, and this ends up inhibiting the interferon pathway. There's a couple other ways HCV can antagonize host cells, but the main takeaway is that it can block the production of interferons and sometimes ISGs as well. Next slide, please.
So interferon therapy is a treatment that is given to patients by injecting them with a type of interferon into the host to combat the virus. So when someone is under interferon therapy, the first step of interferon pathway is skipped, which is the production of interferons into the host cell. The injection of interferons into the host provides a quicker way for the production of the ISGs because only interferon signaling is needed to occur, which allows for the stimulation of ISGs. Also, with the interferon therapy, this will ensure the production of ISGs, even if the virus has a way to evade the first portion of the interferon pathway. The reason why interferon therapy is useful is because it bypasses the production of interferons and can go straight to the binding of IFNAR and then the production of ISGs. This means that it also bypasses some of the antagonisms that hepatitis C has against the interferon pathway. Next. And in this slide, we have a list of the current FDA Food and Drug Administration therapeutic agents that are used to treat patients with different diseases. So in this table, interferon therapy has been used to treat hepatitis B, hepatitis C, human papillomavirus, multiple sclerosis, as well as other diseases. We can see in this table that different types of interferons are used to treat different viruses. The types of interferons that have been used for hepatitis C are those that are boxed in blue, listed under generic name, which happen to be different types of alpha interferons. Next slide. So as I said in the last slide, different types of interferons are alpha interferons that are used to treat different viruses. An example of an interferon that is used to treat hepatitis C are PEG interferons. In interferon therapy, PEG interferon therapy is often combined with ribavirin to fight against hepatitis C. Some of the things to know about PEG interferon treatment is that the treatment will lead to sustained virological response rates of 40 to 45% of the cases. The treatment also gives interferon a longer half-life, which is the amount of time it stays active before it is eliminated. And a small fact to note is that when synthetic interferons were first introduced, they were not pegylated and had to be dosed 3 times a week. But now pegylated interferons are dosed once a week. Next slide.
Yeah, so as Daniel said, interferon therapy ensures the production of these ISGs to help fight HCV. A couple of these ISGs are RIG1, which activates the RLR pathway and inhibits HCV replication. There's also an RNase L, which cleaves HCV mRNA and stops the translation of viral proteins. ISG20 inhibits the replication of HCV replication through 3' to 5' exonuclease activity. There's IFIT1, which targets the IRES-mediated translation and replication sites. IFIT-M can block the virus from entering by disrupting the interaction with the co-receptor CD81. And then finally, overall, there's the production of IRS7, which upregulates the expression of all of these interferons and ISGs. Next slide.
So there's a wide range of possible side effects that one can experience while using interferon therapy to treat hepatitis C. These side effects vary in severity and duration and can have a significant impact on whether patients adhere to treatment and how their dosage is determined. These side effects can be broken down into 4 main categories: bone marrow depression, flu-like symptoms, neuropsychiatric side effects, and autoimmune conditions. Some of the new therapies which are currently being tested and recommended for some patients include protease inhibitors and NS5A inhibitors. Protease inhibitors treatments aim to target the non-structural proteins involved in viral entry and replication. Protease NS4A is involved in viral entry, and NS3 is involved in replication. The pros of this treatment is that it doesn't involve the innate immune system so directly, so it doesn't interfere as much with people who have compromised immune systems, and it can be used as a treatment for hepatitis C for those individuals. And the cons are that this treatment plan is very difficult to adhere to because it involves taking approximately 15 pills per day for at least 4 weeks. Because this is such a difficult plan to adhere to, many people stop their treatment prematurely, which could result in even worse side effects. The second alternative to interferon therapy is NS5A inhibitors, which work to target the NS5A replication complex. This type of therapy has similar pros and cons as protease inhibitors, but while protease inhibitors target viral entry, NS5A inhibitors target viral replication. So one reason that the side effects of interferon therapy are so significant is because they result in 10 to 20% of patients discontinuing their treatment plan prematurely and 20 to 30% requiring dose modifications due to the severity of their side effects. Another accessibility issue, as mentioned earlier, is that interferon therapy often interferes with the therapies used to combat HIV and other immune suppressing conditions, which could result in adverse reactions. And so future therapies aim to be more inclusive of these groups. A third issue with accessibility is that treatment can be very expensive for those who do not have health insurance. So this includes a lot of young adults and those who are unemployed. Also, insurance providers often do not cover the entire cost of treatment unless patients meet certain criteria, which excludes a lot of patients. But we are working towards a more accessible future for hepatitis C treatment through different fee assistance programs led by different interest groups and pharmaceutical companies, and also a more diverse array of alternative treatments for hepatitis C, as we mentioned earlier. And these are our references. And that is our presentation. Thank you.